Right guys, we have got fish gills and gas exchange going on right here. This is a quick diagram up here to help you understand what is going on as I'm teaching you this stuff basically. Right, so here we go. We've got the gill filaments. Gill filaments are here. Well, first of all, actually, I will explain why we're doing this. So gas exchange. We want to make gas exchange as efficient as possible. Oxygen uptake needs to be as efficient as possible. And that's pretty simple. That's just basic, we should know that. So, first of all, we've got the gill filaments. The gill filaments are here. 1, 2, 3. They look a bit like, I don't know, French baguettes or something like that. And here is the gill arch. This attaches the gill filaments here. All the gill filaments do is they increase surface area. Very simple. Part of Fick's laws. All of these things are to increase the fusion rate. As I said, oxygen uptake. Make gas exchange most efficient as possible. So first of all, the first component of the gill structure is gill filaments. Increase surface area. The next component is lamellae. Lamellae, they are adjacent to the gill filament. This means they're 90 degrees to the gill filament, and they also increase the surface area, but we need to know a bit more than just increasing the surface area for the lamellae. They are one cell thick. We know with Fick's law that one cell thick means they have a short diffusion pathway. This means that the diffusion rate is increased again. So we've got increased surface area and increased surface area for both gill filament and lamellae, but one cell thickness, short diffusion pathway, done. But there is one more thing. They've got lots of blood capillaries around them, like a good capillary network around the lamellae. This means that it has an increased diffusion and also an increased surface area. So that is pretty simple. Gill structure out the way, gill filaments and lamellae, that's all you really need to know. Gill arch here, increased surface area, one cell thick, and blood capillaries. 3 things. Done. We're moving on. We're going to go to how they actually use this. How do they actually breathe? 2 seconds. Right. How do they actually breathe? Ooh, that's a bit... I'll bring this down. Right. So, the way they breathe is the countercurrent system. And this means that blood flows in one direction, water flows in the other direction. A key word in the AQA exam is opposite directions. That's it. Now what this does is it maintains a large concentration gradient. That's 1 mark in an exam. If it's asking a basic question about gill filaments, you need to remember that one. That one is key, just like the rest of them were key as well. So maintains a large concentration gradient, sorry. And that's 1 mark. The next mark is maintains a large concentration gradient across the whole length of the lamellae. Another mark. 2 marks already. So across the whole length of the lamellae, just to reassure you. Right. That's it for the countercurrent system. This is to also make gas exchange the most efficient. Now, an exam style question is coming up now. Why is the system only one way? Why does the blood and the water only flow in one way? Like, why does the water flow in one way and why does the blood flow in one way? Well, simple. It's less energy. In our body, we have multiple different systems, and that uses up a lot of energy. Well, in fish, they're only small and they're always swimming around and all this, so it uses less energy. So there has been a question about pressure. I've seen it before. It's not actually in the AQA biology book or the CGP biology book either. This is for OCR as well. There may be some more stuff with OCR or EdXL, but this is the key information that you need, like basic stuff, shortened down to the tiniest time ever to get everything in. So basically the pressure, so it talks about how does the fish make water flow over its gills. The way it does this is we know that the water goes in the mouth and out the gills. That's how they breathe. In the mouth, out the gills, done. So the mouth opens to let the water in. The floor of the mouth, which is underneath the tongue, drops. This increases the volume within the mouth and decreases the pressure, because we know more volume with the same amount of water molecules means there's less pressure. Then the mouth closes, which keeps the water in the mouth basically, and the floor rises back up. This decreases the volume within the mouth and increases the pressure. So now we've got less space in the mouth, but we still got the same amount of water molecules, so that's increasing pressure. This forces the water over the gills. It pushes the water over the gills, and that's simple how you breathe. How they breathe, sorry. Done. Right, moving on. Oh, we just straighten this up. God. So, these are more questions which are like, they're not actually in the book, but they have come up in AQA. So something about temperature. So why do fish take in more oxygen or more water flow over gills in the same time when the sea temperature is increased? So basically, why do fish breathe more when the sea temperature is higher? And this is because there's more enzyme activity going on. If the body is hotter, like just hotter, and there's increase in enzyme activity and increase in metabolic reactions, and therefore increase in respiration. And that's it, simple. We need more oxygen to cater for all of the metabolic reactions that are going on. Now, there has been another question about size. Why is it that when baby fish are just born, they have no gills when they're born, but why is it they can still breathe? Now, the reason is they've got a large surface area to volume ratio. And we know that surface area within Fick's law means that diffusion rate is increased and oxygen uptake is increased and easier. So that's 1 mark. This is a 2-mark question. So large surface area to volume ratio, and they've got a short diffusion pathway because the skin is thin. That is in a baby fish. Short diffusion pathway because the skin is thin. Done. Finished. Thank you for watching. That was fish gills and gas exchange. You just need to remember gill filaments, lamellae, surface area, 1 cell thick, capillaries, countercurrent system, maintains large concentration gradient across the whole length of the lamellae. And done. These questions are more application questions, and they will just come in time, hopefully. Done. Thank you for listening. See you next time.